Hi, everyone. Welcome to Microbial Minutes for June 18th, 2018. I'm Julie Wolfe of the American Society for Microbiology, and it's been a minute since our last Microbial Minutes, and that's in part because we at ASM have been really busy preparing and hosting our annual meeting, ASM Microbe, which was held from June 7th to 11th in Atlanta, Georgia. It was an amazing experience with all kinds of sessions held on just about every aspect of microbiology that you can imagine. And so rather than highlight what's been going on in the news and scientific journals the way that we normally do in these microbial minutes, I'm going to talk about some of the really cool microbiology stories that I heard or heard of at ASM Microbe, so that whether you were there and didn't get to see all of the different sessions, or whether you weren't able to go at all and want to know what happens at this really amazing meeting, you'll have a feel for what kinds of science is presented. So we're going to touch on a number of different fields within microbiology. Of course, microbial sciences touch just about everything. So if you can think it, there's probably a session that's held on your subject at ASM Microbe. I'm going to start off with clinical microbiology, though, since this was the very first presentation that was given during the opening session. The first speaker, Sharon Peacock, gave a talk emphasizing infection control practices, where she compared standard culturing techniques used in hospital surveillance to sequencing approaches, and in particular, whole genome sequencing approaches. She advocated for these sequencing approaches over the standard outbreak detection, because the sequencing approaches can actually identify isolates that are missed by standard surveillance. And she used an example of an MRSA outbreak, methicillin-resistant Staphylococcus aureus outbreak that was found by her own group. This is done in part because blind sequencing of these surveillance isolates does not target only the suspected isolates, but can also detect things such as cryptic transmission, such as when this MRSA is transmitted between family members. And not all family members have overt disease, but they can still transmit it to other vulnerable populations or people who might then come down with a Staph aureus mediated infection. She also compared pipelines of standard outbreak detection to sequencing techniques, and so that using these automated pipelines, which will provide the sequencing technology or sequencing results, as well as the identification of those isolates, can help people to more quickly get data that will help infection control specialists in their decisions. And that although it can be expensive to implement some of these sequencing pipelines, the ability to identify and more quickly quash any potential outbreak will drive down the cost of sequencing versus standard techniques when one considers all of the resources and time that it takes in order to maintain or close down an outbreak using standard surveillance techniques. We also heard about antimicrobial stewardship. Of course, clinical microbiology encompasses all sorts of different fields, but we've talked quite a bit about antimicrobial stewardship on these microbial minutes, and I thought this was worth highlighting because there was an excellent talk given by Lori Hicks, the director of antimicrobial stewardship at the Center for Disease Control and Prevention. She looked at the importance of antimicrobial stewardship to patient safety, and she started by defining what antimicrobial stewardship is. There's actually a lot of different definitions within the field, and what she emphasized is that the CDC likes to measure and improve prescriptions to ensure that patients get the right dose, the right drug, and the right duration of treatment when an antibiotic is needed. And the ultimate goal is not to decrease the number of drugs that are used just for the sake of decreasing drugs, but to improve patient safety and patient outcomes, which is really the ultimate goal of patient care providers. So who uses these antibiotics? As I mentioned, she was highlighting outpatient use because this is 80 to 90% of the total volume of antibiotics used in the United States. The total cost of $33.2 billion. These are prescriptions that are filled by people who might go visit a primary care physician and then go someplace like their local pharmacy in order to fill that prescription, that antibiotic prescription. And this compares with 16.4 billion or 6.5 billion worth of antibiotics used in hospitals or long-term care facilities. Now, within the outpatient use, 30% of these prescriptions are thought to be inappropriate or unnecessary. And it's the goal of the CDC to reduce this, at least within the United States, by half by 2020, which is quickly approaching. That year is quickly approaching. Hicks emphasized that education of primary care physicians or patient care providers is not enough, and that focusing with sociologists and public health workers on changing the or addressing the social and emotional factors will lead to changed behaviors. And she used this CDC public service announcement as an example. Now, I'm going to switch to show you just a couple screens of this. I won't be able to have the sound, but we have a link below. So feel free to click out and then click right back if you want. But this particular PSA shows the mom using the wrong tool to prepare her kid's lunch. And it uses humor as a way to address and explain to people, using an analogy of something that they would understand, something like making sandwich or doing chores around the house, with something that they might not be as familiar with, which is the difference between bacteria and viruses, and which can be affected by antibiotics. All right, so we're going to move on and talk a little bit about environmental microbiology, which was also addressed in the opening session by the second speaker, Veranda Montgomery, who told the story of Freimayella, which is a cyanobacterium that can detect light using a bacterial receptor, a light receptor called RCAE. And then depending on the wavelength of the light that's detected, the cyanobacterium can produce a red or a green pigment. And actually, based on that wavelength, the cell can also have a different morphology, a different shape. This can be an elongated or a more rounded cell shape. And Montgomery called this phenomenon photomorphogenesis. The discovery of black-colored mutants, which actually produce both red and green pigments, bypass the need for this RCA light detection. And this mutant has been able, this and other mutants have been used to dissect the influence of the different wavelengths, and eventually used to discover that the carboxysome size, or the available carbon within the different cyanobacteria, may also be related to light regulation. And so this was highlighted in a paper that was published in mSphere, which I believe we have linked below if you want to read the entire story for yourself. Moving on within environmental microbiology, the first plenary was, one of the plenaries, was begun by Rebecca Vega-Thurber, who also studies environmental microbiology, but focuses on the microbiology of coral reefs. She began by talking about the crystal clear water that we all associate with coral reefs and helps make them such a beautiful place to go scuba diving. But this crystal clear water doesn't have a lot of nutrients for the animals and the many diverse organisms that live within this biosphere. And this is why corals play such an important role. Corals, an animal themselves, rely on their symbionts, which are autotrophic dinoflagellates that can use photosynthesis to produce some of those foundational nutrients that the corals will then use in their life systems. And then this can provide a foundation for many different organisms, that the crustaceans and the fish and all the different things that we associate with the extremely diverse coral reefs. Now, her study is specific to the coral microbiome, and she studies all types of different microbes, but her talk focused on the bacterial microbiome. The role of bacterial microbiome is not very well known, although it's pretty well established that the coral species, every coral species has its own specific bacterial microbiome. And researchers know this because if they look at the different 16S ribosomal RNA genes associated with one coral species, you can see a completely different signature than in coral species that are immediately next to that species, given their species uniqueness. Now, these bacterial microbes are thought to influence the ability of the coral microbes to adapt to different stressors, things such as different types of nutrients, pollution, and temperature change. Vega-Thurber focuses a lot on the temperature changes that are associated with global warming. And she is one of the researchers who is spearheading the Global Coral Microbiome Project, which has so far collected over 3,700 DNA samples from around the world, trying to understand the coral microbe relationship. Now, corals are not very complex organisms, but they do have different parts of their animal cells. They have the mucus layer, which is the outermost layer, the tissue layer, which is right underneath that, and then below that is the calcium skeleton of the animal. And the diversity of these bacterial microbiomes of the coral is actually most diverse within the mucus. And so studying both healthy and unhealthy animals, Vega Thurber and her associates came up with the Anna Karenina principle. So Anna Karenina is a famous tome in which all happy families are alike and each unhappy family is unhappy in its own way. And Vega Thurber Labs said that this is very similar for coral microbiomes. All happy microbiomes are alike and each unhappy microbiome is unhappy in its each way. And this means that coral species that are stressed, for example, by nutrient deprivation, are going to have a different change to their microbiome than coral that are stressed from temperature changes or that are stressed from infection. So all of those different microbiomes will change based on whatever that specific stressor is. And this really mirrors what has been found for the human microbiome, where many human microbiomes can be known to be the same, but then different stressors such as smoking, such as a poor diet, such as lack of sleep or exercise, will have a different effect on the microbiome constituents of that individual. And I should mention that if you want to hear more about coral microbiomes, there was a live podcast recording of This Week in Microbiology, which I have pictured here on the right-hand side. Here we have the panelists. We have Michael Schmidt, Michelle Swanson, Elio Schechter, and the host, our host, Vincent Rocaniello. They interviewed Christina Kellogg, who is also a coral microbiologist and goes into some very fantastic places trying to study the bacteria and other microorganisms that make these animals healthy and or sick. And we have that linked below as well if you want to download or stream the episode. Now, in this same plenary, talking about climate change and microbes, Virginia Pitzer gave an interesting talk about how one might predict the impacts of climate change on infectious disease. Now, of course, in order to make these predictions, you have to consider what the climate projections are. How is the climate predicted to change? And what is the incidence of disease in the given climate? So how, as the climate moves, the disease incidence may move with it. But of course, it's more complex than that, and the change in infectious disease must be predicted to occur at the right time, in the right place, in the right direction, with a hypothesis that includes a mechanism for explanation of that particular change. She mentioned many different examples of variables that can affect some of these models. You could look at pathogen variables, such as the transmission ability of influenza, which is influenced by the absolute humidity of the environment. You can look at host variables, such as the innate immune response of people to rhinovirus, which depends on temperature. And she mentioned a number of other variables as well, things such as exposure, which can change given that a global warming mediated flooding event may bring microorganisms in close contact with people in places where it may not have been previously. She also emphasized that models are extremely helpful, but they cannot predict everything. And one important variable that they do not consider is interventions. So malaria is a classic case that is predicted to have expanded based on expansion of the mosquito vector that carries the malaria parasites. But there have been large efforts in treating and preventing malaria, and the incidence of malaria has actually gone down despite changes with the global vector spread of the mosquito vector. And so it's important to remember that human interventions may mediate or mitigate some of the effects of climate change on infectious disease. All right, moving along, kind of in the more global health sense, we can't talk about global health without highlighting the keynote address speaker, Anne Schuchat, the assistant director, the deputy director to the Center for Disease Control, who highlighted the importance of learning from the past to prepare for the future. She also highlighted technologies such as genome sequencing, like Sharon Peacock did, to help identify the source of outbreaks, using an example of a South African outbreak of listeria that was aided by genome sequencing to pinpoint the source of the outbreak. She also urged the audience to do away with the cliche that disease is just an airplane ride away, and use that as a measure to kind of wash one's hands of predicting where an outbreak may occur. And in this case, she used the 2014 Ebola outbreak as an example of something that was much more difficult than expected, and highlighted that this is a lesson for every country in being prepared, given what they may expect or not expect to encounter. She emphasized that mistakes can be frustrating and humiliating, and how we as humans react to them is what makes the difference, and that it's important to be humble and remember that we're living in a microbial world. And although the keynote address was unfortunately not taped, you won't be able to watch that. If you watch this space, there will soon be an interview with Anne Schuchat, hosted by Jennifer Gardy, an excellent science communicator, and they'll touch on many of these different global health topics that Schuchat has experience with. Also in global health, one aspect that might be overlooked by people in Western societies is the spillover that occurs from the intermixing of people and animals carrying zoonoses, zoonoses being pathogens that reside within an animal reservoir but can also affect people. And this happens with bushmeat. There was an entire session on the spillover of human infectious pathogens from bushmeat, and one of the speakers was Joram Buza, who wanted to look at the pathogen prevalence of these pathogens across Tanzania, where Buza is from. And so with a collaboration with Penn State, Buza and colleagues developed surveillance methods, PCR surveillance, and microbiome analysis techniques training for participants who would go out and gather different samples to isolate and look at what types of microbes are inhabiting those different bushmeats. And so in this case, they got over 37 isolates from different times of the year, because that may influence what the animals what isolates the animals are experiencing, excuse me. And so wet and dry seasons, they looked at 3 different regions from across Tanzania. They looked from more than 12 different species of bushmeat, and both fresh meat, freshly killed, and processed meat. And within their studies, they identified 69 samples that were positive for different zoonotic pathogens, including anthracis, brucella, and coxiella, which were equally distributed across the regions. But they did see patterns that correlated with seasonality, finding more positive samples in the wet seasons and more positive samples in the fresh meat. And this gives public health workers an area to concentrate on when looking for these potential pathogens. Also in the global health vein, but moving more into biotechnology, the third and final opening session speaker, Manu Prakash, spoke about the different types of low-cost tools that his lab has developed. And this was a very inspiring talk, as Prakash has made a number of very inexpensive and yet effective tools that can be used for diagnostics, for disease ecology, and to foster science education across the globe. One of the devices that he highlighted was a $20 flip phone that can identify the different acoustic signatures associated with mosquitoes. And this is important in regions where there's a lot of mosquito-borne disease. You want to know whether the mosquitoes that you might find in your house are potential carriers of malaria or Zika or other types of human disease, or whether the worst that you might experience is a bad rash. And he found that you can use a $20 flip phone to differentiate the hums that come from mosquitoes, different mosquito species, to differentiate which species may be around. He also highlighted the handheld centrifuge that his lab has developed, which is based on an ancient toy that works by pulling some strings apart. And this simple motion can help this handheld centrifuge reach up to 120,000 rotations per minute, which is huge. And this is important for doing things in diagnostic technologies, such as separating blood into plasma and serum. But this energy that's generated can also be used for other applications. And his lab is actively looking at how this can be applied to charging cell phones, such as those that might identify the mosquito acoustic signatures. And finally, he spent a lot of time talking about the Foldscope, perhaps the most famous invention from his lab, which is a low-cost paper-based assembled microscope that can help aid in diagnostics. For example, it helps to identify malaria parasites that might be in that patient sample that has been separated by centrifugation. But he really focused on the ability of the Foldscope to bring the joy of science to a huge global community. His lab has mailed out over 450,000 Foldscopes to over 130 countries. And Prakash highlighted the work of a single teacher in India who has passed out over 55,000 of these low-cost microscopes with really inspiring videos of the children and adults who are inspired to explore the world around them, come up with their own experiments, their own protocols, and just really dive into the microbial world. Also in biotechnology, one of the President's Forum speakers, Hema Reguera, talked about electromicrobiology. Her talk, Microbes to Power, started by telling the audience not to be afraid to talk about electrons. Restless electrons support cellular life. And her studies with Geobacter, a bacterium that oxidizes iron, which we are familiar in the form of rust, is able to do so through the work of its many cytochromes that can transfer electrons to the minerals. And her quest began by asking, how do these Geobacter cells get rid of the mineral after they've respired, after they've passed off their electrons? What happens? Do the cells simply die? And what she and her lab were able to find is that the pili that transfer these electrons will depolymerize and then build up again in order to transfer those electrons elsewhere within the mineral. And with the help of many collaborators, she and her lab have found that the pili themselves are nanowires that are conductive regardless of presence of either the mineral or the bacterium. And this has led to a new program at her institution on bioelectricity. It's led to the generation of many patents and a company that she has spun off, which invigorates trainees who are interested in entrepreneurship and are not necessarily interested in following the academic path of their advisors. And this leads us into microbiology education, which has a number of very interesting sessions at Microbe. The sessions began with Misty Thomas, an educator at a primarily undergraduate institution, or a PUI, who wanted to talk to her fellow professors of PUIs about how she is able to teach 4 classes, sit on committees, act as an advisor, and also find the time to do research. And so she showed what her schedule looks like and how she had protected time each week that was dedicated and protected for research with her undergraduate research students. She used the classroom to test drive students by introducing lab skills such as pipetting or running an agarose gel. So they're familiar with a lot of these techniques by the time they get into the research lab. And these can be used for ready-made projects, things like the Small World Initiative, which we've highlighted at American Society for Microbiology. We have a link to a blog about an entire course based around using discovery-based research and the Small World Initiative to run a lab-based course. But she also said, feel free to create your own research projects. If you do so, simply make sure that you bring the tools that you'll be using in your research into the class curriculum. For example, if you want to have the students use CRISPR to mutate E. coli, give them some hands-on CRISPR experience in the microbiology lab. An important part of undergraduate research is the ability of the instructor to delegate and trust students. Students will inherently make mistakes as part of the learning process, but it's important that they earn your trust in order to feel the freedom to explore further into their research project. And Thomas also highlighted the money and space issues that are ever present at PUIs. She advised the audience to choose your projects wisely and share resources such as space with the teaching lab that can be co-opted for research projects, or money that might be spent on research on lab materials for lab courses that can also work, that can also be used in some of the research projects themselves. Another excellent talk on microbiology education was given by Sarah Brownell, who talked about how do we assess course-based undergraduate research experiences, or cures. There are lots of good outcomes that have been associated with using cures. This is discovery-based research. Students have a stronger sense of science identity, a greater sense of independence, and this has been shown against cookbook microbiology lab courses, those that simply take a protocol that's done year after year to teach students about microbiology concepts. But what is it about the cure that leads to this outcome? What part? Is it the design? Is it the actual research themselves? And Brownell highlighted that the fact that backward course design that begins with course goals is used in many different types of courses, mostly lecture-based courses, but that this will integrate some of the pedagogical goals of the instructor from the beginning if you start with what it is you want the students to experience. This is important to consider because there are often tensions that exist between pedagogical and research goals. For example, in order to publish, students need to generate many different data sets, and so it might benefit the research instructor to have students repeat the same method over and over. But in terms of pedagogy, it might be better to expose students to a wide variety of methods so they understand how these can be applied in different settings and what types of questions those can answer. And which of these is important to focus on can be determined if you set your goal ahead of time. Measuring success is also important. A lot of cures have a presentation, a poster presentation or a talk as the ultimate course ending. And in this case, success is important. It's important for the students to be able to say, we discovered or found this thing. But success is also not always representative of the scientific process, and something that is important to convey to students who are interested in a research career is the potential for an experiment to fail. Now, I want to talk about some things that were not necessarily in these more formal presentations. All the talks that I've discussed so far are representative of the sessions that are held at ASM Microbe. There are a number of different types of talks. There are rapid fire presentations, which are 5-minute presentations given in different lounges that are held throughout the convention center. So these are going on simultaneously, and these are much more informal, more discussion style. Where you can see a presenter will have a television screen where they have access to show a few pieces of data, but then there's a lot of Q&A between the audience members and the person who's presenting. There was also a very fun session called Upgoer 5, which I think of as extreme de-jargoning, since the participants are only allowed to use the 1,000 most common words in the English language to describe their research projects. And so this will lead to descriptions of gastrointestinal disease as sometimes the bad cells make a person do wet number 2s, a very fun and innovative way to think about diarrheal pathogens. Within the exhibit hall, there are not only the exhibitors and the poster sessions that are given by poster presenters, but there are the track hubs, and there are 8 different tracks that are held at Microbe so that every community can find those that are interested in the same subject. And these hubs will host sessions where a speaker, such as yours truly, will discuss concepts such as either something scientific discovery. There can be sessions that are held on publication or science communication or career career tips for applying to different jobs, all types of different activities. And on the other side of the session is a fireside. Every track has its own fireside, which was host to fireside chats. Here I am showing the intimate nature of the folks gathering around the fireside to either listen to Journal of Clinical Microbiology Editor-in-Chief, Alex McAdam, or ASM President-elect Michelle Swanson, hosting discussions on things such as the state of the journal or women in microbiology. Again, the topics are extremely numerous, and it's a great way for people to have in-person discussions with folks who are really influential in the field. Within the ASM booth, you still can't escape all the science that's going on. The ASM studio sessions covered a range of topics each day, had a number of different panels, recordings, et cetera, that would touch on topics such as global health. There were a number of live podcast recordings. I mentioned the TWIM earlier. There were also TWIV, This Week in Virology, and a Micro Talk recording. There were sessions held on science communication tips and tricks, career and CV advice. There was a session on using art as a science education tool, and then some fun sessions on microbiology myths and what's supported by the science, and the science of influenza. Now, these sessions were all recorded before a live studio audience. You can see there was often standing room only in the images that I've collected, and these were taped. So they will be put onto the YouTube channel in the following weeks. So if you're interested in getting notified for these, go ahead and subscribe to the ASM YouTube channel, and these will pop up into your inbox once they're available. Otherwise, check back. They will be coming shortly. Finally, throughout the rest of the exhibit hall, there are all kinds of fun microbiology activities. It's impossible to escape the popularity of Johnny Chang, our resident button artist, who will make a button to the liking of whoever is waiting in line. So those that study coral microbes might like a little scuba diving microbe, such as this one here. And really, the sky's the limit with Johnny's creativity. This was the first year that the ASM hosted an outbreak exhibit as part of ASM's sponsorship with the Smithsonian National Museum of Natural History, where there's also an outbreak exhibit. And so the panels that were hosted at Microbe highlighted the different pathogens that are going to be featured in that museum exhibit. And then every year there is something from the Center for the History of Microbiology Archives, or CHOMA, where ASM archivist Jeff Carr will put together some topic from the archives. And this year focused on anti-vaxxers, but not those that have been boosted by the false claims of associations between vaccines and autism, but those that have had anti-governmental and anti-public health sentiments since the onset of vaccines, with pamphlets from the 1800s that were trying to convince folks that variolation, one of the very first types of vaccination against smallpox, was actually a danger to the public. That's going to wrap up most of the important things that we have time to cover today. Of course, in a half-hour session, we only have so much time. There were so many different amazing scientific presentations. If I wasn't able to get to yours, leave a comment. Let us know what it is that you want to hear. And we will resume the next time our typical microbial minutes, where we discuss the scientific news and journal articles that have been released in the weeks in between our sessions. So look forward to talking with you next time when we resume microbial minutes.